Good afternoon. My name is Giuseppe Legname. I'm a doctor in philosophy, graduated at Warwick University in the UK. I'm a biologist by training, and I'm currently a professor in a university in Italy. The university is called Scuola Internazionale Superiore di Studi Avanzati in Trieste. My current research interests are in neurobiology, and in particular, I'm interested in understanding the molecular mechanism of neurodegenerative diseases. And we are using several models to study this, cellular or animal models to study these diseases. I've been working in particular into the field of prion diseases. I used to collaborate with the Nobel laureate Stanley Prusiner. He won the Nobel Prize in 1997 for his discovery of prions. And lately we've been involved in more research on Parkinson's disease and Alzheimer's disease. So the most exciting outcome of our research in the last few years is the understanding of the molecular mechanism of all neurodegenerative diseases. And what we learn is that despite what everybody else was thinking, is that all these diseases may be related to protein aggregation that eventually becomes infectious and can basically spread from neuron to neuron, from cell to cell in the brain, and eventually cause the disease. This is a novel paradigm regarding these disorders, and in particular it's very important because despite several decades of research, for example in Alzheimer's disease, where very little has been achieved in terms of treatment, therapy, cure, we believe that having this new understanding of the disease may eventually lead to better therapeutic interventions. One particular area that we are interested in the last few years is to basically identify new ways of diagnosis these diseases. And the reason for this is that previously it has been believed that we could develop a drug or a cure even if the disease was at the terminal stages. And this is not true, because once the neurodegenerative disease is at the terminal stage, it's basically irreversible. So understanding when the pre-symptomatic phase of the disease, when the disease starts, is a key for developing more active therapeutic intervention. And this is exactly what we are doing. In collaboration with some colleagues in Italy that work at the University of Verona, we are developing techniques that are taking peripheral nervous system cells, for example, from the olfactory bulb and olfactory epithelia, and ask whether or not in these cells there are protein aggregates, which are basically the first molecular events that leads to this disease. And by studying these processes early on in the disease, we may be able to identify novel and more active ways of halting these disorders. Is it okay?
Mm-hmm. Are there parallel studies that are going on throughout the world?
Yes, there are studies here in the United States, besides, of course, Italy and Europe, and Japan. In general, I would say that all the Western world is playing a very active role in identify any of these new diagnostic tests or treatment. And we are focusing, of course, in the more important range of population, which is the over 65-year-old population. The incidence of these diseases in this segment of population is highest.
You mentioned that the technology and sequencing the DNA in Japan is very sophisticated.
Yeah, well, we are working in collaboration now with the Riken Institute in Tokyo, Wako City in particular, in Yokohama, where they've developed excellent facilities for high throughput sequencing, deep sequencing of the genome. And what we're learning with this technology is that, contrary to what we knew in the past, that was basically the genetics of how an individual is made up, we're now discovering a different layer of complexity of the genome itself. If you remember, a few years ago, there was the sequencing of the human genome, and it was believed back then that having sequenced the genome would have ended basically all the diseases we know. But basically, what we learned from there is that only a tiny fraction of the genome encodes for the classical so-called genes, proteins. And the vast majority of the genome was instead named as a junk DNA. But what we're learning now is that this so-called junk DNA is actually coding for other entities without gene by itself, which do not code for proteins, but regulates the levels of proteins and how a cell develops, and eventually how a cell specialized to be a muscle cells, a nerve cells, a skin cell, and so on and so forth. And besides this, what we're learning now is that there are elements throughout the genome that are rearranged early on in the development of an individual that actually create the individual. Our genes come from our father and mother, but how we are is coming from the rearrangement, the overall rearrangement of the genome. And this is a very recent research that eventually can and will revolutionize the way we see how the individual can resist or basically be more susceptible to diseases.